Welcome to the session 2 of today we're all talking about the scary part, toxicology. Okay, and so we have a speaker, Professor Barbro Melgert. She's from the University of Groningen, Netherlands. She's currently working as the Associate Professor at the Department of Molecular Pharmacology. I guess you have heard what we're talking so far. Okay, so you know what she's working on it, and she's working on the plastic, microplastics, and also lung diseases. So let's welcome Professor Melgert to make a presentation on the inhalable textile microplastic fibers and their effects on the lungs. Professor Melgert, stage
Thank
yours.
you very much. I'll
share your screen.
my screen.
Yeah.
There you go.
Yeah, proceed.
All right. Well, thank you very much for inviting me to talk about our study into inhalable microplastics and effects on their lungs. Very happy to talk about these results to you. So something I don't have to tell this crowd, I guess, is that plastic is a problem at the moment, especially plastic pollution. And a lot of our attention has gone to the pollution of our waterways, so the seas and the rivers and all the junk that ends up on beaches. But microplastics are also in the air. So there are a lot of fibers and particles in the air made of plastic, especially degradation products of tires and synthetic clothing. And these microplastic particles are irregularly shaped. Most of them are fiber-like because they're from synthetic clothing, or shard-like. And of course, they range from very small, like nanometer size, to bigger particles. And whether these particles in the air could be a problem for our lungs depends on their size. So I just wanted to take you through our lungs and what happens with different sized particles. So these are our lungs, and we have conducting airways that transport the air to the more peripheral parts of the lungs, which consist of air sacs, really tiny, small structures. And what we know is that we can breathe in particles, and most of them will be caught by the mucus in our nose, but particles that are smaller than 100 micrometers, so around the size of the diameter of a hair, they can get stuck in our airways. And the smaller these particles get, the deeper they can get. So in order for a particle to get to the air sacs, it has to be between 1 and 5 micrometers. If it's bigger, it will get stuck in airways. And if it's smaller, it might stay suspended in the air and you might actually breathe it out again. When we design therapeutics for inhalation, we actually make sure that the particles of these inhalation products are between 1 and 5 micrometers. So size matters for these microplastics to get into the lungs. And why are we interested in these microplastics in the air? That's because there have been some studies that show that the indoor levels of microplastics are much higher than the outdoor levels. And most of our time is spent indoors. So there's a lot of opportunity for exposure for a lot of people. And we also know that an average house accumulates around 20 kilos of dust per year, and that around 30% of that dust consists of synthetic fibers. So there is around 6 kilos of microplastic dust in our homes that we could potentially breathe in. And that might have an effect on our lungs. There are a few studies that looked at whether there are microplastics in lung tissue. So the first one is an older one, was around 20 years ago, in which they looked at lung tissue from patients with lung cancer, and they studied both the tumors and the healthy tissue around it. And they found actually synthetic fibers in 100% of the tumors, and in around 80 to 90% of the normal tissue around it, they also found synthetic fibers. And then a recent study looked at lung tissue of people who died of non-respiratory causes, and they also found fibers or particles in this tissue, which was more distributed throughout the lungs and in the alveolar or the air sac lung tissue. And the most common particles they found were polypropylene and polyamide. So the other piece of evidence of interactions between microplastics and lung comes from the textile industry. We know that people working in nylon flocking, so that is making of fabric that sort of has this velvety feel. So what they do is they make little tiny nylon fibers and they glue them onto a surface fabric. And these factories contain a lot of these nylon fibers in the air. And we know that 30% of the employees in these industries develop respiratory symptoms, inflammation of the airways and of the more peripheral tissue. So there appears to be an interaction between microplastics and lung tissue, and not in a good way. But we know very little about what causes these problems in the people in the textile industry. So what we wanted to know, what happens when the cells of the lungs that first come into contact with microplastics actually come into contact with them. And these cells are epithelial cells and macrophages. So epithelial cells are the cells lining the airways and the air sacs, and macrophages are the immune cells in the air spaces that are there to help the epithelial cells to fight off things that we breathe in. So think viruses, bacteria, but also particles. When you breathe those in, macrophages will be the first cells to try and dispose of these inhaled dangers. So in order to really investigate these interactions between these cells well, we used reference textile microfibers that we made ourselves. So we ordered nylon and polyester threads from Goodfellows, and we made very standardized fibers from these threads. And we use polyester and nylon because these are the most common plastics used in clothing. And for polyester, we ended up with fibers that were around 15 by 45 micrometers, and for nylon, 10 by 30 micrometers. And we use these in a model of lung organoids. And so these are basically sort of mini lungs that develop from primary lung cells and develop into structures that look like tiny lungs. And we do this by first taking lung tissue from either humans or mice, and we isolate epithelial cells from these from this lung tissue. And these epithelial cells are then cultured in a sort of a gel-like material and grow out to structures that resemble lung tissue. And these are some examples for you. So this is a tube-like structure that resembles an airway and is also coated with epithelial cells that we find in the airways. And this is a grape-like structure that resembles the air sacs and also is coated with epithelial cells that we find in air sacs. And we use this model to test what these microplastic fibers could do. And here you can see examples of the organoid cultures with and without the fiber. So this is the control condition. You see these really big structures, tube-like structures, which are the airways. And then with the cyan arrows, you see the tiny little structures that are more grape-like and are the air sacs. And here in these two conditions, you see lots of fibers for one. So these rods are all the fibers. And you can also see that there, especially in the case of the nylon, there is hardly any outgrowth of these organoid structures. So we did not find any airways in this well, and only a few air sacs. And for polyester, you also see some inhibition, but not as profound as for nylon. And we quantified this. So here we have the airway organoids, and these are the air sacs. And what you can see here is that nylon really inhibits the outgrowth of airway organoids. Polyester seems to have a tiny little effect. And the effect for the air sacs was also there, but not as profound as for the airways. So we use this model also for human cells. We get lung tissue from people who undergo a lung resection because of lung cancer or transplantation, and we isolate epithelial cells from these from the lung tissue of these patients. And we use the same method of organoid culture, but the organoids that grow out from human lung tissue are not as defined as the ones from the murine tissue. So we don't actually see these airway and air sac organoids, but we see sort of a mixed phenotype with both types of epithelial cells being present. So that's why there's only one graph here. But what is obvious is that in the control condition, we see outgrowth of lung organoids. But if the lung organoids were allowed to grow with nylon, there are less organoids forming. And the effect for polyester, again, is less profound. So then what we wanted to know is the way we set up the experiment is that we incubate the cells that we isolate together with the fibers in the gel from day one. And there could be, so is this effect that we see, is that a direct interaction of the fibers with the cells, or is it something that is leaking from, in this case, the nylon? Because of course, all these microplastics contain lots of additives that might be leaking out and causing this effect. So in order to investigate this, we redid the experiment and we cultured the cells as we did before with the fibers in the gel. But we also had a condition in which we added the fibers on top of the gel. So there was no direct contact between the forming organoids and the fibers. And we also made a fiber wash and added this to the medium in which the organoids are cultured. And what we found was that, again, when the nylon fibers were inside the gel, there was profound inhibition of the outgrowth of organoids, but also when the nylon fibers were on top of the gel or when we added nylon wash. And this suggests that it's not the fibers themselves that have this effect, but it's something leaking from the nylon. And then, of course, next question is, what is leaking from the nylon? And for that, we analyzed the nylon wash to see if we could find which compounds may be causing this. And so this is actually what we found in the nylon wash, is that there were many monomer, dimer, and trimer cyclic nylon derivatives present in this wash. So when nylon is made, when the monomers form this long chain that nylon is, some of these monomers can also cyclicize and form shorter compounds, monomers, dimers, and trimers. So we isolated these cyclic oligomers from the wash, and we treated the organoids with these compounds. And so here you can see the monomers, the dimers, and trimers, and we also used a combination of these oligomers. But they didn't appear to have any effect on the outgrowth of organoids. So even though they were present in high concentrations, these were not the culprits for the effect we saw. So we're currently further investigating to see what it is that is causing this effect, and we haven't figured it out yet. So more to come. Then another question we had was, so this was sort of an effect of fibers on a developing organoid. But if you're an adult and you breathe in these fibers, then of course your lungs are already developed. So would these fibers also have an effect on already developed organoids? And to do this, we changed the model a little bit. So we first had the organoids develop from day 0 to day 14. And then we added the nylon fibers on top of the gel, or we added the nylon fiber wash. And we had the organoids growing for another 21 days to see what would happen to their size and their development. And this is a bit of a complicated graph, so I'll take you through it. So we had two controls, just the cells without anything added, growing for 14 days and for 21 days. And you can see there's a nice number of organoids developing, and there's no difference between 14 and 21 days. Then we had a positive control in which we had the nylon fibers added to these cells from the start of the experiment for either 14 days or 21 days. And again, we see that this profoundly inhibits the outgrowth of organoids. And then in two other conditions, we first had the organoids growing for 14 days without anything being present. And then we added the fibers on the gel, or we added the nylon wash. And then you see that there's actually no effect of nylon on the organoids, not on the number of organoids or on their size. So this suggests that nylon, or whatever is leaching from nylon, is particularly affecting the developing organoids. We did RNA-seq analysis to see what is actually being changed by the compounds leaching from nylon. And this is a really complicated process. So our airways consist of different types of airways with different types of epithelial cells. So the larger airways have different types of epithelial cells from the epithelial cells that are present in our air sacs. But all of these have epithelial cells that have a sort of a stem cell-like character. And this is actually what we use in our organoid model, is these stem cells within the lungs, those are the cells that grow out to these organoid structures. And what we found after the RNA-seq analysis is that the compounds in nylon really change the development of the epithelial cells and the certain developmental pathways, but that's a bit of a detailed story for this presentation. So that's epithelial development, but of course, epithelial cells are not the only cells that are being exposed to fibers that you can inhale. It's the immune cells, the macrophages, that also see those fibers first. And we wondered if we incubated lung macrophages with nylon fibers, what they would do with it. Would they just ignore them, or would they start to try and eat them? So in this experiment, this is in vitro, we labeled the macrophages with a green dye. So the macrophages are the green dots, and we labeled the nylon fibers pinkish red. And we followed the macrophages to see what they would do with these fibers. And as you can see in this movie, is that the macrophages do not ignore the fibers. They really try and eat them, and they even seem to be fighting over them. So whenever you inhale a fiber, a macrophage will try and do something with it. So this was a shorter time frame. But here there are some pictures of what happens when you look at the longer time frame. Again, here this is a control condition with the cells being the cytoplasm being stained green and the nuclei stained blue. And here the fibers are stained pink. And after 1 hour, you don't actually see that the cells have eaten any fibers, but you do see some interaction of the cells with the fibers. But after 18 hours, some of the fibers actually end up in the cells. And the cells really need to grow to be able to contain these huge fibers, because the fibers are generally about twice as big as the cells. And what we further showed, and what I won't show you now, is that these macrophages also get activated when they eat these fibers. So they start producing inflammatory mediators, which could then be responsible for the inflammation seen in the lungs of people that inhale nylon flock, for instance, in the flocking industry. So to summarize, macrophages eat fibers and get activated. Exposure to nylon or polyester microfiber inhibits organoid outgrowth, and developing organoids seem to be particularly affected, and that's mostly by compound leaching from nylon microfibers, and this leachate affects epithelial development. And if you want to look at these results in more detail, we put our manuscript on bioarchives, and please have a look if you're interested. Implications of this is that developing or repairing lungs may be most at risk for effects of microplastics, or at least of nylon. So children and babies, but also people with a lung disease, because these people have damage to the lungs and they need to repair this. So there's constant epithelial repair in the lungs of people with a lung disease, which might be impeded by inhaled microplastics. But this is all only relevant if we inhale large numbers of these fibers, and that is something we don't know. We don't actually know how much is suspended in the air in our houses and how much of that we breathe in and how much is retained. So that needs to be assessed to be able to really determine what the hazard of microplastics is to our lungs. I would like to conclude with thanking all the people who have contributed to this study, particularly Francine van Dijk and Shanshan Song, who did most of the work for these studies. And of course, our funder, the Dutch government, who really thinks that we should have more info on microplastics and is funding lots of projects looking at microplastics in human health. I thank you for your attention.
Thank you, Professor Barbro. It's a nice presentation. I'm Ramanathan from India.
Hello.
Okay, the nice presentation, and because we all don't know that our cloth and fibers, the nylon, will be creating so much problem, and also the leaching out of that is also creating problem. There are some few questions for you. You will have it one or two, huh?
Okay, that's fine.
Yeah. What is your view on the toxic effect of natural fibers when compared to synthetic fibers?
Oh, that's a very interesting question. So we know that people from, for instance, the cotton industry also develop respiratory symptoms when exposed to high levels. So it could be that it's just anything you breathe in is not good. But we actually haven't compared natural fibers versus the microplastic fibers. That's something that we really want to try and see if there are any differences there. But anything that is irritating to the lungs usually leads to problems. And we're not sure if natural fibers are better than the synthetic fibers.
Added to that, there are also people exposed to the complex like air pollution and asbestos when they move out of that. So those will complicate the situation, or?
Yes, definitely. So, and we'd hope that asbestos, of course, is a diminishing problem, because everybody's now aware that asbestos is not good for your health. But air pollution, yes, that's actually something we looked at in our project too, to see, because we know that microplastics can really absorb other toxicants. So we did some experiments in which we exposed microplastics to diesel exhaust, or to cigarette smoke, to see if they would absorb some of the chemicals from these air pollutants. But in-depth analysis didn't really show significant amounts of particles or compounds attached to the polyester and the nylon microfibers. So for now, that seems to be a minor problem, but of course it might depend on the type of plastic you're looking at. Maybe some other type of plastic may absorb more of the air pollution, and there might be interactions. Or there might be interactions of inhaling them both at the same time, right? So that's something we haven't looked at yet.
Okay, that's right. I think you have answered this one. Do you have any input on how macrophages could have damaged or removed nylon? That I think you have already discussed.
Yeah, I think they're trying to, but I think they cannot, because they cannot digest it, right?
Okay. So thank you very much. I think there is one more question that you can answer it online. And that is
Okay.
what is the instrument? Thank you very much.